But we're actually very lucky and very happy to have Shankar with us, the CEO of OCGN, presenting the company. I've known Shankar now for close to 2 years, and his team, they've been focused on gene therapies to cure blindness and disease, and especially in the last few months have really also kind of jumped on the COVID-19 vaccine area, the space, with the view of being of adding to vaccine sort of supply out there. They've also, I think, been putting out some decent bit of news flow out there from them and their partner, I believe, Bharat Biotech, Shankar. So with that, we'll let you take away, give us the presentation, and please leave us 5 or 10 minutes for questions.
Great. Thank you, Hartaj. Thank you for having me. Good morning, everyone. Again, OCGN is a biotechnology company. We're focused on developing gene therapies for curing blindness diseases and working on a COVID-19 vaccine to save lives. Starting with Covaxin, which is COVID-19 vaccine, it's based on a whole virion or virus. It's an inactivated vaccine with adjuvant, and it's licensed from Bharat Biotech in India. And currently, Bharat already has an emergency use authorization, and they've actually vaccinated in mass immunizations or millions of people, and it's safe. So recently, we have announced efficacy from the interim analysis. It's 81%. It looks very promising, and they also have generated significant immune responses in the phase 2 clinical trial, both humoral as well as cellular. Cellular response are very important for long-term durability of any vaccines. Again, the phase 3 clinical trial includes about 25,800 patients. And we're planning to, and again, the salient features of the clinical trial, what they have done is it included a 12 plus age group. Currently in the US, all the 3 EUAs, which they have approved, and they cover 16 plus, and 2 of them cover 18 plus age group, and 1 of them covers 16 plus, but there is a significant children population which we are not focusing on, which is very important to control the pandemic. And I'm going to talk more about it a little later in the presentation. Another thing which is very important is because of this broad coverage from this whole virion vaccine, we're able to show it's effectively neutralizing UK variants. And that's very important because, you know, there are a lot of variants, and the virus keeps escaping, and we get more variants everywhere. So it's really important to control that, to control the pandemic. And I'll go into more details a little later in the presentation. Now, coming on to our OCGN's gene therapy platform, we believe it's breakthrough and game-changing technology for ophthalmology space. Again, we got 2 programs in that, OCU400, based on the NR2E3 gene. Recently, we got broad orphan drug designations for retinitis pigmentosa and LCA. And again, you know, these 2 broad indications cover about 175 genes with mutations. So we believe our one product has potential to treat all these diseases. And the next one on the list on the gene therapy is OCU410. It's based on Aurora gene, and it has a potential to treat dry age-related macular degeneration, which is a huge disease burden. About 9 to 10 million patients in the US alone struggle with it. Currently, there are no approved therapies globally. And it's a huge disease burden, unmet medical need, and we are planning to put this program in the clinic next year. We also have a very strong strategic partnership with a large biotech company, Consino Bio, for gene therapy manufacturing, which is really paving the way for us to move into the clinic very rapidly. And we do have a novel biologic in the preclinical stage. It's progressing well, targeting DME, diabetic macular edema, DR, and wet AMD. And once again, because of the distinct mechanism of action, we believe this can also target non-responders to current VEGF therapies. Again, we're planning to bring this program to the clinic next year. We do have a very strong leadership team with a proven track record of success in developing, launching, and managing life cycle of many biopharmaceuticals successfully. And we got a very highly esteemed scientific advisory boards, one for the retina and one for vaccine scientific advisory board. Once again, with licensing of COVID-19 vaccine, and we put a very, very highly solid scientific advisory board, 3 people from University of Pennsylvania, including Susan Weiss, who is a coronavirus expert, to David Fagenbaum, who is a top immunologist, and he runs the cytokine lab, cytokine storm lab at Penn, and he leads FDA panel and NIH panel for repurposed drugs for COVID-19. And then a lot of industry folks like Satish, Bruce, and Kathy coming out of the strong vaccine background. Bruce used to run all the clinical trials for vaccines at Wyeth and Pfizer. So we do have a very strong a team really working on the vaccine as well as on the ophthalmology side. Coming on to pipeline snapshot, once again, as I mentioned before, the Covaxin is in pre-commercial stage right now, and we're waiting for a little more data from our partner in India so that we have a regulatory path, and we're waiting for some more data. And once we get that on the safety and efficacy, we'll be able to file for UA in the US. And then our goal is to commercialize it as soon as we get the UA approval. And then going on to modified gene therapy program, OCU400 has received 4 orphan drug designations for different mutations from FDA. And as I mentioned, we also have broad orphan drug designations from EMA. I mean, our goal is eventually to get a broad indications. So we are planning to file IND for NR2E3 and rhodopsin mutations this year in the US by the end of the year. And then we can move on to the early stage clinical trials. And again, OCU410 is in the preclinical stage. Our goal is to push it into the clinic by next year. Similarly, OCU200, which is a transferrin tumstatin fusion protein targeting major retinal diseases such as diabetic macular edema, diabetic retinopathy, and wet AMD. It's a significant disease burden again, about 10 million patients in US alone. And there is a significant underserved disease population, non-responders to existing diseases, and also people looking for, disease-modifying drugs. So we hope, we can help these patients. We're working, very hard on this program too, so that we are able to, expecting to put this in the clinic next year. So with that, I will dive into more details on some of the programs, starting with Covaxin, which is licensed from Bharat Biotech. So this is, again, a product profile. The proposed indication is active immunization to prevent COVID-19. The target population, again, we do have the data from our clinical trials. We're going to target 12 plus age group. This is very important. Currently, none of the companies are covering this with existing EUAs. And if you take like a 12 to 16 population, I mean, that's a significant, you're talking about at least 16 million kids in the middle school and high school, which can be covered with this. And so then the dose levels, again, it's 2 doses, 0 and 28 days. 28 days is a booster. And it's going to be presented in a multi-dose vials because that's easy for, mass immunizations. One of the, and another key features in this is it's stored at refrigerated temperatures, which is very important. And it has a, predicted shelf life of, a couple of years. And it's also stable at room temperature. So for mass immunizations, when you have to store it on and off, and it can minimize any losses. It's easy to distribute and easy also for stockpiling. If the pandemic is under control, CDC always stockpiles for future breakouts. And this would be ideal vaccine for that instead of, vaccines which are stored in frozen conditions, which is very difficult to store for the long term. So why Covaxin? When you have 3 other vaccines in the marketplace? That's a very good question, right? So again, it's current vaccines are all based on a spike protein, and Covaxin elicits broad spectrum immune responses against multiple antigens and multiple epitopes, which is very important. And again, we have shown it effectively neutralizes UK variant, and potentially it can neutralize other variants too. That's a big plus. And again, it's easy to stockpile, as I mentioned before. And it's also based on a proven technology platform, inactivated viruses. Many vaccines were developed using this platform. It's safe for all age groups, including polio vaccine, which is given to babies even today in the US using the inactivated viral platform. And once again, the phase 2 studies did cover 12 plus age group, which is unmet need in the US currently. And again, the formulation contains novel adjuvant, which boosts Th1 response, cellular mediator response, which is very important for long-term immunogenicity. So as I mentioned before, again, how do you differentiate this vaccine? In the other vaccines, currently there are 3 in the market today under EUA. 2 of them are mRNA, 1 of them is adenoviral based on the right side. They all focus on the spike protein, single antigen. And on the left side, Covaxin has a whole virus. So you got multiple antigens you're targeting, including spike, nucleic acid protein, membrane proteins. That's very important, So when you have a variant, when you have the mutations, you tend to target the spike, then you get a spike mutations, and then you're coming with newer vaccines to target that variant. That variant again mutates, So I think somewhere you need to stop the chain. The best way to stop the chain is coming up with a broad-based vaccine, which can have a multi-antigenic and multi-epitope approach. And that's ideally that can control the pandemic. That's what we have to focus on. We believe, Covaxin has ability to get there. And this again sums it up with all the vaccines, in US, Pfizer, Moderna, and J&J under EUA, including Astra, which is globally distributed. They all are based on spike protein, and Covaxin is based on whole virus, which significantly distinguishes. And this has got EUA in India, and currently it's going through mass immunizations. This again, the technology comparison with other vaccines in the space. Once again, the stability is a key feature compared to some of the frozen vaccines, which is very important for distribution, supply, and also long-term stockpiling. And another key feature is the B and T cell immune responses against multiple protein is very, very important for not only, as I mentioned, potentially to control the pandemic. And safety and efficacy in phase 1-2 studies is very, very, very safe, and the numbers are similar to what you see in the traditional vaccines, under 10%, and the mild to moderate side effects. And once again, this sums up the phase 2 clinical data, which got published in the Lancet recently. So if you look on the left side, you have seroconversion rates. This is after 28 days. 28 days is the second booster. After 28 days, 14 days is 42, and another month is about 56 days. Now you're looking at seroconversion based on a couple of assays, which is plaque assay, PRNT, as well as MNT. In both the cases, you see 98 to 96 to 98% seroconversion, which is pretty significant. And on the right side, another feature with this vaccine, it has got 2 adjuvants, aluminum hydroxide and IMDG, as you can see on the right side graph. And you see a significant increase in the T cell responses to IF and gamma, TF and alpha, and IL-2, which is very important. And again, on the right side, if you look into that, we summed up our phase 3 interim analysis. It's 81% efficacy. And this also covers the analysis up to date over 2,500 and about 2,500 people over age 60. They go all the way to age 91. And also 4,500 people with comorbidities. Again, we believe this vaccine has a potential to be used because of the safety for all age groups and a high-risk population, such as people with comorbidities and pregnant women. And again, we had to do some more studies in the US to get those broad labels. So in a nutshell, this vaccine has shown significant antibody response, neutralizing antibodies, as well as significant T cell response, cellular mediator response that is necessary for long-term durability, and also very good safety. Now, this slide actually here, this is a strategic outlook for Covaxin progress. And on the left side, numerous studies starting from preclinical to clinical. Everything is published in highly credible journals. All the clinical trial data is published in Lancet, phase 1 and phase 2, and some of the preclinical data is published in Nature. And our goal is again, to use the data which is generated in India by our partner, and to get the EUA, And because in addition to the phase 3, they also started mass immunizations. It's already dosed, millions of people in India, which is important, right? Safety is extremely important for vaccines.
Right.
So our goal is to take this and get the EUA, then roll out the vaccine in the US and as early as second quarter. And based on our agreement with Bharat, they're going to provide initial tens of millions of doses. And we're going to actually test the product in the US at one of the FDA-approved laboratories, re-release it to the US market. And then we're also working with US manufacturers to establish manufacturing in the US. Then once the initial rollout is complete, we'll have these US sites ready to up and running, and we'll be supplying doses from those sites. So our goal again this year, we're targeting about 100 million doses for the US market. So with that, now I will spend some time on our modifier gene therapy platform. So how do we differentiate ourselves from a traditional gene therapy? There is one gene therapy product in the marketplace in the ophthalmology space, Luxturna, and that targets RPE65, very specific genetic mutation. So this one is illustrated on the left side of the slide. Here, you got a normal gene. It's a defective gene. So you give a functioning gene through AAV vector. Once you start expressing it, you control the disease progression. So that's good. But if you want to develop, a product or target another mutation like P23H, or something, you have to develop another product again using the functioning gene. It takes about 8 to 10 years to get the product approved with the significant capital and resources. So if you have a disease, as I mentioned before, between retinitis pigmentosa and LCA, 175 genes are mutated. So it's almost impossible for us to develop 175 products and rescue these patients. These patients are in a severe need, and also they need a significant rescue, because majority of these patients do become legally blind by the time they're in mid-40s. So we believe we have a solution for that. On the right side, that's where our modifier gene therapy platform comes in. These are modifier genes. They regulate the expression of many gene networks in the retina and associated genes. So these networks are responsible from a gene, the cell development to inflammation to survival. It's very important. So what these modifier genes do, they regulate these gene networks and associated gene expressions and reset the homeostasis and restore the normal cell function. As a result of that, these almost act like master genes in the retina. And so they have ability to go after many broad spectrum diseases such as RP. So you don't have to develop a product by gene mutations, so that it's gene agnostic. So we have received multiple orphan drug designations because of the data we demonstrated. And so that means it has ability to go after broad disease criteria like RP and LCA, which could be significantly benefit to the patients. So here, again, this is a high-level concept of modifier genes. On the right side, as I mentioned, there are 3 genes in this spectrum, NR2E3, NR1D1, and RORA. And they control different gene networks, as we outlined, like photoreceptor development. And some of the key mutations are outlined in there. Again, this modifier concept is not new. It is already well known in the disease areas such as cystic fibrosis and spinal muscular atrophy, where the modifier genes have linkages to clinical phenotypes. So we are the first company to bring this concept into ophthalmology space. So last year, in collaborating with our inventor at Harvard Medical School, we published a paper in Nature, and that sums up demonstrating the broad spectrum effect of these genes. And taking a single product, OCU400, based on NR2E3, looking into 5 different animal models, early stage of the disease, late stage of the disease, looking into multiple endpoints, including functional endpoint, and showing this therapy offers significant rescue consistently among all these models. And also the therapy is safe. And once again, the Nature publication link is there at the bottom of the screen. So now I'm going to show high-level data from this publication. So here in this slide on the top left, you got early stage rescue in 5 different animal models, treated and untreated. Treated means treated with a single subretinal injection of OCU400. On the right side, again, it's a advanced stage of the disease. This is based on the histology, looking at outer nuclear layers in the retina. At the bottom, what you see are the fundus imaging. Again, all the animal models are named there, RD1 to RD7. Above each of the animal model, that's a clinical, human disease, a phenotype. And RD7 represents enhanced Escon syndrome. So on the untreated and the fundus at the bottom of the image, you see a lot of spotting. That means retina is degenerating. And in the treated animals, you see a significant less spotting. That means there is a significant rescue. Now coming on to the top, outer nuclear layers, on the untreated on the left side, there's a significant rescue, and several layers are left in the treated animals compared to untreated. On the right side, in the advanced stages of the disease, and you still see a significant protection. Typically, if you have 20 to 30% layers left, you should have a good vision, so reasonable, right? And in the advanced stage, you don't see any layers left for untreated animals. So this is very compelling data. Now going on to functional data set, here you got a ERG, electroretinogram on the left side scotopic, which is well-lit conditions. And so you have here, again, you're showing significant increase in the ERG signal from treated versus untreated, which is, again, demonstrating rescue is significant across all animal models consistently. And now this sums up a safety. One could argue, hey, if you're giving a broad spectrum therapy, is the therapy safe? Here, looking into fundus imaging on the left, histology, and looking at the opsins, and immunochemistry, consistently showing the treated and untreated, there's no difference. Again, and the therapy is safe, including ERG on the right side. So here is our broad stroke regulatory strategy. What we are planning to do is initiate 2 parallel clinical trials, MRT-300 opsin, and we're currently completing our GLP tox studies. We already manufactured the product at a commercial scale at 200 liters and successfully done. And our goal is to, again, it's the same product going after 2 parallel phase 1-2 clinical trials. Once we have the data, we believe we'll be able to use that and do multiple phase 3 clinical trials in parallel. Eventually, our goal is to get a broad RP or LCA indications with the potentially, with some phase 4 commitments. So this slide actually sums up comparison with the traditional gene therapy and cell therapies. Traditional gene therapies, as I mentioned before, you can go 1 target at a time, 1 mutation at a time. Cell therapies could be a little broader, but depending on the cell types. Again, there are no approved products in cell therapy. It's still experimental therapy, and they can go like cell types like RPA cells, and they'll be photoreceptors. They'll be subjected to that, but they're a little broader than the a traditional gene therapy. Whereas we can have a very broad stroke treatments using our gene therapy program. Again, this is a bit. Want to spend just a little time on the OCU410 targeting dry AMD. It's a very tough disease. That's why there are no approved therapies to date. Again, there are multiple factors here, right? I mean, one is oxidative stress, inflammation, and lipid metabolism. RORA being another master gene, we believe it controls all these functional networks. Therefore, it could be potential to treat this huge disease. Just imagine treating the disease such as this one, about 10 million patients in the US, tens of millions globally, with a single injection, in the back of the eye. That's the power of this platform. And we're very excited about this product. And we're really working hard to put this in the clinic next year. So we do have a strong partnership with Cancino Bio. That's about $15 billion market cap company. And this is really paving the way for us to rapidly move. Even under the COVID situation, the manufacturing, nothing was impacted. Really thanks to our partners for really actively working on this program in the middle of the COVID and really delivering our product. We're ready for our clinical trials. As I mentioned before, we're waiting for our tox, studies to be completed. Then we'll be filing the IND at the end of this year. Now, I'm going to spend a couple of minutes on our biological program before I close out. Again, OCU200 is very unique. It's a tungsten transferrin. It's a fusion protein. It acts on integrin receptors, which are only present on the active endothelial cells. Distinct MOA. We believe this can target even non-responders. There are about 50% of the patients are non-responders to currently available anti-VEGF therapies, and there is a significant global market. This slide actually sums it up. Head-to-head data with currently approved anti-VEGF therapies which are in the market. Eylea is currently a gold standard. On the left side, in the neovasc model, you can see 10 microgram dose of OCU200 shows pretty good equivalent response to Eylea at 20 micrograms. In the middle, you see a wet AMD model. It's a laser-induced CNV. In there, OCU200 head-to-head is statistically superior to Avastin. Again, on the right side, we did studies to show Eylea and OCU200, it's trending pretty good. And obviously, the combination will have a better impact. And so once again, we believe this has the ability to be a disease-modifying drug because it inhibits new blood vessel formation. It's anti-inflammatory and antioxidative too. So again, summing up the key features of this technology compared to anti-VEGF and anti-integrin, other anti-integrins, there are other anti-integrins which are in the clinical trials, but they are small molecules. However, they have proven the target. They work in the DME and the space. And however, we are the first company with a macromolecule taking it to the clinic. And again, the salient features, unlike other anti-VEGF therapies are, it actually works targeting integrin active endothelial cells, and it triggers a native anti-angiogenic process, and it stops the formation of unwanted blood vessels. That's very important. It's also pro-apoptotic and antioxidative. So based on the studies we have seen, we are expecting the durability is going to be good for this product, and we're expecting that to be, at least 3 months or longer, which is very important for this population. So with that, again, we have multiple inflection points coming up this year and next year, starting with Covaxin. We're working extremely hard, our team and our team of advisors, day and night to bring this much needed vaccine to the US market, the differentiated vaccine. And so our goal is to commercialize this year and continue to move, several of our gene therapy programs from the clinic and our biological. We're planning to initiate in our ophthalmology side at least 4 clinical trials within the next 1 to 1.5 years. So with that, thank you, Hartaj, for giving me opportunity to present OCGN today.
Thank you, Sanjay. I'm sorry. I wish we had some time for questions. I'm sure we'll have in the future, but thank you for presenting and good luck. You've got a lot going on and, just keep it going. Just keep executing.
Thank you so much.
All right, Sanjay.